Mm. Hello, hello. Good morning. Okay. Can hear me, yeah?
Yeah.
Okay. Yeah. All right. So yesterday we were up to the physics unit 1B. Okay. So we are almost finished. Okay. So today we will finish off the workbook questions. Okay. Last two questions. Let's take a look. Okay. So here we have a tracer consists of a plank resting on two supports which exert upward forces of P and Q. As shown, the weight of the plank is 80 Newton. Okay, acting through its center. Okay, take moments about the left support to calculate the value of Q. Okay, so here we actually have two pivots. Okay, so we just focus on the one that the question asked for. Okay, so left support. So we just focus on the left support. Okay, so moments about the left support. Okay, so yeah. Okay, so moment about the left support, we have um one force here. Okay, so we have one force here, 80 Newton. Okay, so we know that moment, okay, um equal to FD. Yeah, okay. So moment equals to FD. So we have this is clockwise. This is clockwise moment. So we have F1. Okay. And then this distance is okay about the pivot is 3 meter. Okay. So this is D1 equals to 3 meters. Okay. Actually, we have this force. Okay. But this force, the D is 0. The distance to the pivot is 0. So this part FD will be equal to 0. So we ignore this one. Okay. So we don't care about this one. And then we have another force, which is this one, Q. Okay. This Q is going because it's pushing up by this pivot. If this pivot is not there, the plank will fall down. Okay. But because the pivot is there, it's got a force pushing up. Okay. So this is the second force. Okay. So for this force, it will be F2. Okay. And then the distance, it will be from here to here. Okay. Between the force and the pivot. So the D2, it will be 3 plus 2. Okay. 3 plus 2, it will be 5 meter. Okay. Just like that. Okay. Now, as usual, it is not moving. It is equilibrium. Yeah. Okay. So when we have equilibrium means clockwise, okay, moment will be equal to anti-clockwise moment. Okay. So this one is anti-clockwise moment. So this one is clockwise moment. So we're going to have F1 D1 equal to F2 D2. Okay. So F1 is 80. Okay. And then the distance D1 is 3. Okay. And then equal to F2. This one we need to find out. F2 D2 is 5 meter here. Okay. So 5F2 is equal to 240. F2, which is Q, which is what we want, 240 divided by 5. So that will give us 48 Newton. Yeah, okay. So which means to support this plank, not letting it fall down, the up force of Q needs to be 48 Newton. Yeah, okay. So if we did everything correctly. Okay. So now we already know that for equilibrium. Okay. So we have no resultant moment. Okay. Because the clockwise moment equal anti-clockwise moment. Okay. We also have another condition, which is, okay, no resultant force. No resultant force. Okay, so which means in this case, the up forces will be equal to the down forces. Okay, so the down forces, okay, is this 80 Newton. 80 Newton, the up forces will be P plus Q. Yeah, okay, P plus Q. So we already know Q is equal to 48. P plus 48 equals to 80. Okay, so now we want to calculate the value of P. P equals to 80 minus 48 equals to 32 Newton. Okay, so that's how we solve this kind of question. Yeah, okay. So maybe we only practice a few questions. Maybe you still don't have too much experience, but later we try to come back to this topic again and practice more. Okay, so now we'll look at the next question. Okay, next question. So we have a gopher, okay. A gopher strike a stationary ball of mass 45 grams, okay. Yeah, so as usual, we just try to change everything to SI unit. 1, 2, 3. Okay, 0.045 kg. Okay, so this is needed for our calculation to have the correct calculation. So we have a person, okay, playing the golf, okay. So he's striking the ball, okay. He's striking the golf ball, okay. Okay, so this is a golf ball. Okay, so we look at what is the information that we have. We have mass 0.045 kilograms. The time, okay, in contact with the ball for a time of 0.001 second. And then the force is 1,350 Newton. Okay. Calculate the acceleration of the ball during the time it is in contact with the club. Okay. Yeah, so for this one, we can use a simple formula based on the information. We can use this one. Okay. F equals to ma. Okay. So we want acceleration A equals to F over m. F is 1350. M is 0.045. Okay. So that will give us, okay, 30,000 meter per second. Yeah. Okay. Yes. Okay. So it seems like, okay, not much the force, but actually the acceleration can be a lot. Okay, 30,000 meter per second squared. Okay, calculate the velocity of the ball just after it is struck. Okay, so yeah, so for this situation, okay, we will use, okay, so what we will use this formula. Okay, Ft equals to mv minus mu. Okay, so basically Ft is equal to change in momentum. Okay, so this is the formula that we can use. Okay, so now F we have, okay, so T we have, M we have, okay, and then V is, okay, final velocity, okay, which is what we want to find, okay, velocity of the ball just after it is struck, okay, final velocity. Okay, U is initial, okay, velocity. So we know initial velocity is 0 because at first the golf ball was not moving. At first it was stationary. Okay. So now we can just input all the information that we have. Okay, F is 1350. Okay, time is 0.001. Okay. M is 0.045. V, okay, minus 0.045 multiplied by 0. Okay, U is 0. Okay, so we can calculate everything. Okay. Okay. So this will give us 1.35. Okay, so this part goes to 0. Okay, 0.045 multiply 0. Okay, so here we have 0.045 V. Okay, so V is equal to, okay, 1.35 divided by 0.045. Yeah, okay. Okay, so that will give us 30 meter per second. Okay, just after the ball was struck. Okay, that was the initial speed. Okay, next. Okay, give two ways which the velocity of the golf ball could be increased. Okay, so how can we increase the golf ball of the speed? Okay, so we can see here, we focus on this formula. Okay, so the speed, it depends on this mv here. So which means, okay, we want this one to increase. We want this one to increase. We want the ft to increase. Okay, because if ft will increase, the mv will increase, so the v will also increase. So if we want ft, okay, to increase, okay, so either the force should be increased or the time should be decreased. So that will give no, and the time should be increased also. So for the force to increase and the time to increase, if both of these increase, then the total of ft will increase. Okay, yeah, so our answer will be uh longer to extend collision time, or we strike harder. So either the force increase or the time increase. So when this one increase, ft will increase. Okay, so that's the concept there. Okay, so now with this, we finish all. Okay, so practice question for this unit 1B. Okay, so finish all. Okay, that's good. Okay, so now this unit 1B, the notes also, we already finished all. Okay, that's good. Yeah, so this one. Okay, so this physics unit 1B is done. Okay, yeah, okay. So now we will move on to the next topic, okay, which is biology. Yeah, okay. So now we will move next topic. Okay. Yeah, so forces and momentum, 1B is done. So we'll move to biology. Okay, unit 2. Unit 4. Okay, chapter 4, biological molecules. Okay, so this is a very short, okay, very short and very simple topic. Okay, so should be quite easy. Okay, I think we can finish today. Okay, let me open it. Okay, so the notes I already sent to you a few days ago, so I think you have it. Okay. Yeah, so you can take a look at the notes at your side also. Okay, so very, very short topic. Okay, yeah, very easy. Okay. Okay. Yes. Okay, so we can take a look. Biological molecules. So means the molecules that are produced by living things. Yeah, okay. So the molecules that are produced by living things. Okay. And then for human, there are a few important biological molecules. So we just focus on human. Okay. The biological molecules are for human mostly. Okay. So for human, mostly we have a few. Okay. Okay. So I'm sure you are very familiar. Carbohydrates, fats, proteins. Okay. So yeah. So they are kind of look like this. Okay. Yeah. So first yeah, and then we'll look at the elements. Okay. So carbohydrate is made of carbon, hydrogen, oxygen. So we know this. They are um they they are they are present in the periodic table. Okay, so these elements. Okay, and then fats and oils, they also made out of carbon, hydrogen, oxygen. Protein, they have, okay, also carbon, hydrogen, oxygen. Yeah, so we know that these three elements are important for living things. Okay, so protein also have nitrogen and sometimes also having sulfur and phosphorus. Okay, yep. Okay, so first we will focus on glucose. Yeah, so we'll take a look at glucose. Yeah, okay. Yes, glucose. It looks like this. Okay, carbohydrates. Okay, so carbohydrates, we have different categories. Okay, yeah. So carbohydrates, we have three categories: monosaccharide, disaccharide, and polysaccharide. Okay, so monosaccharide, we have three types: glucose, fructose, galactose. Okay, so basically their shape is like this. Okay, hexagonal shape, just their groups, their side groups will be slightly different. Okay, glucose, fructose, galactose, they are mostly very similar. Okay, so fructose is a five-membered ring. Okay, like this. Yeah. Okay, but they are mostly very similar. Okay, so disaccharides. Okay, disaccharide is two unit. Okay, two monosaccharide joined together. Yeah. Okay, so monosaccharide, single sugar molecule. Disaccharide, two sugar molecules linked together. So maltose will be glucose plus glucose. Sucrose will be glucose plus fructose. Lactose will be glucose plus galactose. Yeah, okay. Polysaccharide will be long chain, many sugar molecules linked together. Yeah, so when we have many sugar molecules joined together like this is what we call polysaccharide. Okay, yeah. So example, starch, glycogen, cellulose. So these are polysaccharides. Okay, so this example of carbohydrates. Okay, so we can see here glucose subunit will be glucose. Okay, and then example, starch, glycogen, cellulose, sucrose, and so on. Okay, so next, fats. Okay, so fats, the subunit will be made out of fatty acids and glycerol. Okay, so it looks kind of like this. Yeah. Okay, on. Okay, so type of lipid. Okay, so why we have the lipids and the fats? Okay, so it's used for long-term storage of energy in the body. Okay, so it made of a glycerol backbone. Okay, so this part is the glycerol. Yeah, okay, so glycerol with three fatty acid. Okay, so three fatty acid. Okay, yeah, so this one will be the three fatty acids. So it looks kind of like this. Okay, so the long chain is basically made out of carbon and hydrogen. Okay, and then we have two types: saturated and unsaturated. Yeah, so if you see here, saturated means all the carbon will be single bond. Yeah, okay, so this saturated. And then unsaturated means we have some carbon-carbon double bond. Okay, so we have saturated and unsaturated fats. Okay, yeah, so lipids is also called triglyceride. Okay, yes. Okay, yeah, so example of the lipids: vegetable oils, example olive oil; animal fats, cod liver oil from the fish or waxes. Okay, next we have protein. Okay, so protein is made out of subunit amino acids. Okay, so amino acids subunits. Okay, uh, we have 20 different amino acid subunits. Okay, for human. Okay, so amino acid, it looks kind of like this. Okay, so we have the central carbon, and then we have amine group at the side, and then we have carboxylic acid at the side. Yeah, so this is what we call amino acid, and then we have a single hydrogen here, and then this R group, okay, will be different for different amino acid. Yeah, so it depends on the amino acid. So when we have different amino acid, they means they have different R group. Okay, and then what happened? This amino acid can join together, so they can form this peptide bond. Okay, so this amino acid can join together. Okay, like this. Okay, so they join together one by one. Okay, and then they can form this protein. Yeah, so this amino acid joined by the peptide bond, they can form very long chain, and then they can, okay, wound up together to form this alpha helix, and then they can join together more, okay, tertiary structure, and then quaternary structure. So finally, they form these complex protein molecules. Okay, so they start with this simple chain of amino acid, and then they will go through some steps to form these final complex protein molecules that have many functions. Okay, yeah. So example of the protein enzymes, muscle, hemoglobin. Okay, so these are all proteins. Okay, next. Yeah, so carbohydrate, okay, fats, okay. What else? Yes. Okay, so we have these large carbohydrate molecules, okay? Large carbohydrate molecules such as starch, glucose, glycogen, and cellulose. So they are made out of long chain of smaller units of monosaccharides such as glucose. Okay, so we can take a look. Yeah, so it looks like this. Okay, so the type of polysaccharide. Okay, so just now we already said that the polysaccharide we have starch, glycogen, cellulose. Yeah, okay. So it's kind of look like this. Okay, so cellulose, the source is the plant, and then it's made out of beta glucose. And then this one is not too important. Yeah, okay. For IGCSE. So basically they have no branching. So the shape looks like this. Okay. Yeah, so the bonding is look like this. The shape looks like this. Okay, so starch, we have two types. Okay, so we have two types, amylose and amylopectin. Okay, both coming from plants, and then both from alpha glucose. And then the shape is kind of like this. Okay, so the main difference of this starch is one is a straight chain, one has branching. Yeah, okay. And then finally, glycogen. Glycogen is from animals, alpha glucose. Okay, they also have branching. Yeah, okay. So the glycogen in human, it looks like this. Okay, so all of them, they coming from glucose. Okay, next. Yeah, lipids. Okay, so this one we already know. So the lipids, fats, and oils. They make out of one glycerol unit and three fatty acid. Just now we saw that. Proteins is made out of, okay, what we mentioned just now, long chain of amino acid. Okay, so 20 different amino acids in the human body. Okay, yeah. So just now, okay, the sugar molecule, it looks kind of like this. Okay, so when we have glucose, fructose, galactose, so the side group will be slightly different. Okay, yeah. Okay, next. Okay. Yes. Okay. So just now we mentioned in the human body, we have 20 types of amino acid, 20 type of common amino acid. So here we can take a look. So this one, we don't really need to know for IGCSE. So you just need to know roughly, okay? What is going on there, okay? Yeah, okay. So these are the type of, okay, amino acids that we have in our body. Yeah, okay. So basically, This will be the R group. Yeah. So basically, this part is the same for all the protein. Yeah. So this part is the same. Okay. Yeah. So this part is the same. Okay. And then all of them have a different R group. Okay. Yeah. So example, this one, the R group is different. Yeah. Okay. So they have different R groups, and then when they join together, they will produce a different protein with different function. Okay, so it's like Lego. Okay, so we'll join a different Lego shape to get a final shape that is different. Okay, yeah, okay, so it can be very complicated. So these are all the amino acids that we have in the human body. Okay, next. Yes, yes. Food tests. Okay. So just now we already learned about the different type of the biological molecules in the humans. Okay. So mostly in humans and some plants as well. So now we need to know how to test them. Okay. So how to test them. Okay. So the main biological molecules we need to know how to test will be starch, reducing sugar, proteins, and fats. Okay, so I think you know quite well. Suppose already learned year 7 and year 9 until year 9. So this is a quick revision. Okay, so now we take a look at the reducing sugar. Okay, so what it means by reducing sugar. Okay, so we have two types of sugar, reducing and non-reducing sugar. So in IGCSE, we don't need to know too much. We just need to know that we will test for reducing sugar. Okay, so reducing sugar, they will be having a free aldehyde and ketone group. Okay, and then yeah. So example of reducing sugar, glucose, lactose, maltose. Okay, so means our test will be active for glucose, lactose, maltose. Okay, non-reducing sugar, they have does not contain free aldehyde and ketone group. Okay, and then example, sucrose, glycogen, insulin, and so on. Okay, so our test later, they cannot test for non-reducing sugar, only reducing sugar. Okay. Yeah. So now we will look into each test. Okay. So first, to test for the starch. Okay. Test for the starch. We have the starch test. Okay. Starch test. So what do we do? Okay. We will add. Okay. So we will take our sample. Okay. And then we will drop iodine solution. Yeah. Okay. So we'll drop iodine solution. If we have a positive result, it will turn to blue black. Yeah, okay. Okay, let's take a look. Okay, so this is the iodine test. Okay, so we will drop the iodine solution, and then if starch is absent, it will just remain as brown color or yellow color, which is the color of the iodine. Okay, so if starch is present, it will turn to blue black, the color. Okay, so this is how we test for starch. Okay, next. Um, okay, reducing sugar. So how we test the reducing sugar? This is what we call Benedict's test. Okay, so Benedict's test, how we do it. Okay, so first, okay, we will take our sample. Okay, and then we'll put the Benedict solution. Okay, so okay, so we'll take 1 ml of Benedict reagent, and then we will put 2 to 3 drops, okay, into the sample, okay, 2 to 3 drops of the Benedict solution into the sample, and then we will boil it, okay, heating for 3 minutes, okay, boiling for 3 minutes, and then we will see the result, okay. So this will be the result, okay. So if we have no reducing sugar, it will just remain as blue color, okay. No reducing sugar, no color change, it's just blue color. If we have a little bit of reducing sugar, it will be green color. Okay. If we have some reducing sugar, it will be orange color. And then if we have lots of reducing sugar, it will be turn to red color, red precipitate. Okay. Yeah. So basically, okay. So turquoise. Okay. So turquoise will be the original blue color. If a little bit of reducing sugar will be pale green, and then more pea green, and then orange, and then brick red. Okay. So what we call brick red precipitate. So the further the color change, the more closer to red color, the more reducing sugar present. Okay. So this is how we do the reducing sugar tests. Okay, next one. Okay, protein. Okay. So to test for protein, we have the Burette test. Okay. So how we do the Burette test? Okay, so we add equal amount of sodium hydroxide to a solution of the food. So we take the food sample. Let's say we put 5 ml of the food sample liquid, and then we put 5 ml of sodium hydroxide, and then we mix carefully, and then we add few drops of 1% copper sulfate. Yeah, 1% copper sulfate without shaking the mixture. Okay, without shaking the mixture. Okay, what we expect. Okay, so if we have no protein, no protein, it just remain as blue color. Okay, negative Burette test. Okay, if we have protein is present, it will turn to purple color or purple halo. Okay, yes. So this is what we call positive Burette test. Yeah. Okay, so it will turn to purple color. Okay, next. Okay, fats. Test for fats. This is called emulsion test. Okay. So what we do, we dissolve the food in ethanol. Okay. So we add ethanol to the food, and then we pour this mixture, food plus ethanol, into a clean test tube of water, and then we observe it. Okay. Yep. Okay. Yes. So this is what happened. Okay. So we put the food with the ethanol, and then we shake it, and then we add to water, and then if it turns to milky color, okay, milky color, okay, or white emulsion, this one, white emulsion means fat is present. Okay, so if fat is not present, it just remain as colorless. Okay, so if fat is present, we will have this milky color or white emulsion. Okay, so this is the test for fats. Okay, next. Okay, we also have extra one, which is a test for vitamin C. Okay, we vitamin C is important food molecules. We also learn how to test it. Okay, so vitamin C, the structure it looks like this. So the test is DCPIP test. Okay, so how we do it? Okay, so DCPIP is a deep blue color. Okay, so first we measure 2 cm³ of DCPIP solution into a test tube. Okay, and then we will add the fruit juice. We know fruit juice has a vitamin C. So we will add the fruit juice drop by drop, and then we will count the drops used. And then we will shake the mixture after each drop. Okay, so we shake it. Okay, if it's still uh return to colorless, then we will keep adding. If it's remain as blue color, means it's done. Okay, yeah. So if the vitamin C is present, the DCPIP will decolorize. It will turn to colorless. Yeah. Okay. Yes. So DCPIP solution, we add the sample drop by drop. Okay. So if it turns to colorless, means vitamin C is present. Okay. Vitamin C is present. If it remain as blue color, so means no vitamin C. Yeah, okay. So these are all the tests that we need to know for the IGCSE. Okay. Yes, yes. Okay, and then quick note, the food test usually no heating. Only the Benedict test we need to do the heating. Okay. So next we will look at the sample question. Yeah, so we can take a look. Okay, so this sample question. Okay. Yep. Hold on. Okay, so we are asking how you would compare the vitamin C content of a range of fruits. Okay, so let me just copy the. Okay, so you can see this is a 5 point question. It's like a short essay question which can come up in exam. Okay, yeah. Okay, okay. So let's say we have 5 fruits. Okay, so let's say we have like kiwi fruit, orange, okay, mango, okay, lemon, something like that. Okay, so we want to check which one has more vitamin C. Okay, so what do? We will use the DCPIP test. Okay, so first I will juice the fruits. Okay, so we will get the juice. Excuse me, you get the juice for the fruits. Okay, so we'll get the juice. Okay, then I would measure 2 cm cube DCPIP solution into a test tube. Okay, so yeah, so this will be the DCPIP solution. Okay, 2 cm cube. Okay, and then using a pipette, I would drop the juice. Okay, so the juice we will drop into the DCPIP solution. Okay, and then drop by drop. Okay, so we add 1 drop, we shaking, and then record how many drops are needed to make the DCPIP change from blue to colors. Okay, and then I would repeat the test for other juice. Okay, washing the test tube and pipette thoroughly between each test. Okay, the juice that causes the color change with the fewest drop has the highest concentration. Okay, so let's say the lemon, we know the lemon will be having the highest vitamin C. So which means let's say the lemon, okay, it just need 3 drops, okay, to change the color. Okay, but let's say the mango is very sweet, the mango will need 20 drops to change the color. So which is telling us the um the lemon, it has more vitamin C because we just need to put a little bit of lemon juice, just 3 drops, we can change the color of the CPIP. But mango, it just have very little vitamin C, so we need to put 20 drops to change the color of the DCPIP. Okay, so that is the concept there. Okay. Next. Okay. So in this chapter 4, we also have a brief look at this structure of DNA. Okay. Yeah. So this structure of DNA is also very, very important biological molecule. Okay. For the human. Okay. So yes. Okay. So we'll take a look at this. Okay. So the DNA molecule is basically, okay, it's coming from where? Yeah, so we can take a look. Where this, DNA molecule come from? Yeah, okay. So it come from the cell. Okay, so inside the cell, we have the nucleus. And then inside the nucleus, we have the chromosome. Yeah, okay. So each human cell, we have some number of, we have 26, 46 chromosome. Okay, in each human cell, we have 26 chromosome. I mean 46 chromosome. Okay. Yeah. And then if we take this chromosome, if we unwind, yeah, if we unwind this chromosome, we will find this DNA. Yeah, we'll find this DNA. If we straighten it up, we will get this 2 strand. Yeah, we'll get this 2 strand like this. Okay, so these 2 strands, they are joined together by this bond. Yeah, okay. So this is what we call DNA. Okay, so this DNA molecule is made out of 2 strands. Okay, so it is twisted to form the double helix. Okay, so just now we saw the double helix. Okay, twisted to form this double helix. Okay, and then what else? Okay, and then the bases, we have the ATCG, the bases. Okay, we have this pair of bases at the DNA. This is what we call pair of bases, A, T, C, G, 4 types, okay? And then what we need to remember at this point is A and T always together. A and T always together. C and G always together. Okay, just like that. Okay, next. Okay, exam style question. Yes. Okay, exam style question. So, okay, what we need to do, make a table for carbohydrates, fats, and protein with the heading shown below. Okay, so which means, okay, we need to write down for carbohydrates, fat, and protein. Okay, we need to know the chemical elements present, the subunit, and the example. Okay, so this is quite straightforward. We can remember that, no problem. Okay, yeah, okay. So carbohydrate, okay, made of carbon, hydrogen, oxygen. Subunit is glucose. Example, starch, glycogen, cellulose, sucrose. Okay. And then for the fats made of carbon, hydrogen, oxygen. Okay. Subunits, fatty acids and glycerol joined together. One glycerol, three fatty acid. Okay. Example, vegetable oil like olive oil. Animal fats, cod liver oils and waxes. Okay. Protein made out of carbon, hydrogen, oxygen, nitrogen, always, sometimes sulfur and phosphorus. Okay, subunit will be amino acids, about 20 different forms in the human body. Okay, and then example of protein, enzyme, muscle, hemoglobin, cell membranes. Okay, yep. Okay, next. Second question. Okay, student was testing a sample of food for the presence of protein and reducing sugar. Okay, state the name of the reagent needed to test for protein and reducing sugar. Okay, yeah, so which means we need to memorize, okay, for protein and reducing sugar, what is the reagent needed? Okay, so that's easy, we know that. Okay, so for uh okay, burette reagent and sodium hydroxide solution. Yeah, so basically we need the burette reagent for the protein. So what's burette reagent? Is sodium hydroxide plus 1% copper sulfate solution. Okay, so this is burette reagent for protein. Okay, for reducing sugar is Benedict solution for reducing sugar. Okay, next. Which test involve the boiling? Which one need the boiling? We know that. That is the uh Benedict test for the reducing sugar. So this is the one that needs boiling. Okay. Next. Okay. Yes. Okay. So this is the last question. Okay. Part of DNA molecule has the base sequence of this one, ATT, CGACAG on the strand. What will be the sequence basis on the other strand? Okay. So let's say we have one strand, which is, okay, ATT, CG, okay, ACAG, okay. So which means the other trend will be, okay. So A is always with T, T is always with A, C is always with G, G is always with C, and then A is always with T, C always G, A is T, C, G, okay. So we expect the other strand will be this one. Okay, TAAGC. Okay, TGTG. Okay, let's check our answer. Okay, TAAGC, TG. Okay. Okay, maybe we make a mistake there. Okay. Yeah. So the last one is G. Okay, so this one will be C. Okay, so that's our correct answer there. Okay, so we just need to remember A and T always together, G and C always together. Okay. State the name of the structure formed when two strands of DNA coil together. Okay, so when two strands of DNA coil together, what we call that? Okay, we call it double helix. Okay, straightforward. How the strands of the DNA are held together. Okay, how they hold together, okay, is because of the um, bonds between the pairs of bases. Okay, so we have this bond between the pair of bases that hold them together. Okay, yep. Okay, with this we finish. Okay, so we finish all of this unit 4 biological molecules. Okay, that's good. Okay, yeah, so which means here is done. Yeah, biological molecules for the textbook part is done. Okay, so now we will move on to the workbook. Okay, so the workbook is also very straightforward. Okay, yeah, okay, so we can try together. Okay, so this one should be very easy. You can try. Okay, so list the chemical elements that are present in the carbohydrates. Okay, so you can try. You can tell me the answer. For carbohydrates, what elements are present? Okay, you can look at the notes if you want. Yeah, you can try. Okay. Okay, tell me your answer once you have it. Okay, what kind of elements present in carbohydrates? Okay, what kind of elements present, okay, in the carbohydrates?
CHO.
Correct. Yes. Okay. Carbon, hydrogen, oxygen. Yep, yep. Okay. Let's.
Also CHO.
Mhm. Okay. Proteins.
CHNO.
Yep. Excellent. Okay. Next. Okay. So we have this on the diagram. We need to label. Okay. So this one we know straight away. This one will be the glucose subunit, and then the bond will be chemical bonding, okay, between the glucose subunit. So what do we call this? Okay, so part 1, okay, we need to give the name. Part 2, we need to tell the use. Okay, what is it used for? Okay, so part 1. Okay, so this one could be starch, could be carbohydrate, okay, could be cellulose, okay. The function, okay, what is it used for? To obtain energy, okay, because it is a storage of energy. Okay, how about this one? This one we know, okay, straightforward. We have glycerol and fatty acids, okay, and then fatty acid and glycerol join together by chemical bonding, okay. Okay, example, fat, lipids. Okay, so what is it used for? Used for energy. Okay, so it is an energy storage, insulation. Okay, formation of cell membranes, hormone formation. Okay, very useful. The next, okay, so we know this. This is protein made up of amino acids joined together by chemical bonding. Okay, yeah. So what is it? It is protein used for growth, tissue repair, enzyme, used to make cell membrane, and also hormone formation and so on. Okay, so proteins are very, very useful. Okay, next. Yes. Okay, so this one, we need to draw a line to match the statement to the food test. Okay, so this is easy. Okay, yeah, so this one you can try. Okay, so vitamin C, what is the chemical reagent, and then what is the color? You can refer to your notes if you want. Okay, so what is the chemical reagent for vitamin C?
ECPIP.
Mm-hmm. And for positive result?
Colorless.
Yes. Okay. Reducing sugar?
Um. Benedict.
Yes. Yes. Okay. Benedict solution and positive result?
Brick red.
Yeah. Okay. Fat?
Ethanol.
Okay. And positive result?
White emotion.
Okay. Protein?
Be red.
Yes. Positive result?
Blue. Black. No, purple.
Yes. Okay. So finally, starch will be using iodine and blue black for positive result. Okay. So excellent. Okay. So this topic is easy. Okay. Yeah. Okay. So now, okay, this diagram shows two strands of a section of DNA with the bases on the left strand identified. Complete the diagram by writing the letters of the bases that would be present on the other strand. Okay, so this is easy. So this one you can try. So what we expect to see the bases on the other strand. Okay, you can try. So for G, we will be matching with what? Okay, so what will be the bases present on the other strand? Okay, you can try. Yeah.
CA.
Mm-hmm.
G.
Mm-hmm.
TTG.
Yes. Okay, excellent. Okay, that's great. Next, what is the name given to describe two strand coiled together? Okay, you can try this.
DNA.
Close. Okay, double helix. Yeah. Okay. So, yeah, as we mentioned just now. Okay. Yep. Okay. Here. Yeah. Okay. Yep. So DNA, when coiled together, it will form this double helix. Okay. Okay. Next, this is a calculation question. Okay. 28% of bases in a DNA is base A. Okay. Calculate the percentage C of C bases. Show your working at each stage of the calculation. Okay. So we are saying that we have, okay, 100%. Okay. So 100% of DNA. Okay. So, uh, like this. Okay, hold on. Okay, like this. Okay, so 28% A. Okay, so which means it will be 28% T, right? Okay, so now we take 100% minus 28% minus 28%. So that will give us 44%. 44%. Okay, so which means this part, okay, so this part is 56%. Okay, 28 plus 28. So this part 44%. Okay, so which means, okay, C and G, it will be equal. 22%, 22%. Okay, so that's how we work out how much of the C bases. Okay, yep. Okay, so final answer. Okay, 22% of the C bases. Okay, so this type of question sometimes come up in the exam. Okay, and then next. Okay, describe how you would carry out a food test on a piece of fish to find out if it contains oil. Okay, so we know we are testing for oil. This is straightforward. We will be doing emulsion test. Okay, so first we will mix or grind the fish with ethanol. So we just cut the fish into like a paste. Okay, so we make it into a paste, and then we put inside ethanol. Okay, and then we will filter the mixture to remove the fish, the fish, the flesh of the fish. So we'll left the oil in the ethanol, and then we will pour the filtrate into water. Okay, so we'll pour into water. Okay, so this will be the steps. Okay, what result will indicate a positive test? Okay, we know that positive test will be a milky white emulsion. Yeah. Okay, next. Which two statements about DNA are correct? Okay, which two statements are correct? Each strand contains chemicals called fatty acid. Okay, no. DNA has no fatty acid. T always pair with A. Yes. A gene is a length of DNA coding for a protein. Yes. So this one we didn't mention, but that is the case. Okay. A gene is a length of DNA. Okay. So example, okay. So we will take one part. So this is what we call a gene. Yeah. So if we take up one part, this is what we call a gene. So this is maybe for the gene for the color of the eye, okay, or color of the hair, okay, something like that. Okay. So we have a gene for each and everything. Yeah. Okay. So we have a gene to make a hemoglobin. We have a gene to make the carbohydrate make a glycogen, something like that. Okay, so we have a gene to do everything. Okay, yes. A red blood cell contain DNA in nucleus? No, the red blood cell has no nucleus and no DNA. Okay, so our answer will be 2 and 3. Okay, so our answer is C. Okay. Yes. Next. Okay. Outline the structure of DNA. Okay. So this is our DNA here. Okay. So how can we describe this structure in words? Okay. So sometimes it can come out in the exam. Describe this structure in words. Okay. So how can we do that? Okay. So this is what we can say. Okay. So this is a 4-point question. Okay. So first we say that it's made out of 2 strands. Each strand is a chain of nucleotides, okay, the bases. Reference to, we have the presence of bases, and then we have 4 types of bases, A, C, G, T. And then we have bonds that hold the pairs together. And then A always pairing with T, C always pairing with G, okay? So these are the points that we can include, okay? Yeah, and it is done, okay? So this is a short topic, so we finish all, okay? Practice questions for this unit 4. Okay, so it's done. Okay. Yep. So this unit 4 is totally done. Okay. So now we will move on to unit 5. Yeah, okay. So now this unit 4 already done. Okay, so now we'll move on to unit 5 of biology. Okay, so biology, we have more topics and then they are shorter. So yeah, we will do more of them. Okay, so next we will move on to unit 5, which is also quite short. Okay, so we will go through that. Okay, let me open. Okay, unit 5. okay, of the biology and the workbook. Okay. Okay, so see whether we can finish it today. Okay. Yep. So it's very easy, very straightforward. Okay. So this topic is about enzymes. Okay. So enzyme is very important molecule in our body. Yeah, very, very important. Okay. So what is enzyme? So before we talk about enzyme, we'll talk about catalyst. Okay. So what is a catalyst? So you might have learned in chemistry before. It's a substance that increase the rate of chemical reaction. Okay. So basically it will speed up the chemical reaction. It will make chemical reaction go faster. So this is what we want. We want chemical reaction to go faster so we can get the product faster. Okay. And then another important characteristic is not changed by the reaction. It just remain the same. Okay. After the chemical reaction. Okay. So what's enzyme? Okay. So enzyme is a type of catalyst. It's biological catalyst. Okay. So enzyme it help to speed up the chemical reaction in our body. Okay, so that's enzyme. Okay, so we can take a look at the notes. Okay, yeah, okay. So most of the chemical reaction that happening are living things, they are helped by the enzymes. Okay, so enzymes they act as catalysts. They help to speed up the chemical reaction. Okay, next the name of enzyme. So the name of enzyme like lipase, protease, amylase, something like that. Okay, so they almost always ending ase. Okay, so this is the name of the enzyme. Okay. Um, yeah. So enzymes, they usually will speed up the reaction, but sometimes they will slow down the reaction also. Okay. So the enzymes, they used to build up molecules like glucose. We need enzyme to join the glucose together. We also need enzyme to break down the glucose. Okay. So yeah. So both process we need the enzyme. Okay. Okay. Example, starch phosphorylase. Okay. We use the starch phosphorylase to join the maltose to turn it to starch. Okay. So we can see how it happened. Yeah. Okay. So you see, okay, this is the maltose molecule. Okay. So it's made out of two glucose. Yeah. So these maltose molecules we can join together to make starch. Yeah. Okay. Or we can break down this maltose to get the glucose. Okay. So both ways can happen. And then you can see for different process, we will use a different um, enzyme for it. Okay, next. Okay, and then we have another example, breaking down molecules. Break down molecules. Okay, so we can use protease to break down protein to amino acids. Okay, yep. Okay, so we have this protein molecule. We add the protease. Okay, so basically this is our digestion process. We break down, we use the protease to break down the protein molecules to get the amino acid. Yeah, okay. Next. Okay, so enzyme molecules, they are basically proteins. Yeah, each will have active site with a special shape that the substrate can fit into. This is called a complementary shape. Okay, so once the enzyme molecules and substrate come into contact, the products will be formed. Yeah, okay. So this is what we're talking about. Okay. So each enzyme, okay, it will have a special shape. Yeah, the active site, the special shape that is for the substrate to fit into, and then it will form the enzyme substrate complex, okay, enzyme product complex, and then finally it will form the product. Yeah, okay. So this is a breakdown process. So break down this substrate into two product. Okay, so it's very specific. Okay, so only this one can fit into. So let's say we have like this. Okay, so it cannot fit. Okay, or we have like this, it cannot fit. So we are saying that this enzyme is very specific. Only certain things can fit inside. Okay. Another key point here. Enzymes and temperature. Okay. So the enzyme is only active at certain temperature. Okay. So it normally have an optimum and best temperature. Okay. So is around 37 degrees Celsius, which is our body temperature. Okay. So if we increase the temperature above the optimum, okay, so if the temperature is too high, okay, the reaction will slow down. Okay, because the enzyme molecules will denature. Okay, so what it means by denature? Yeah, this is the situation. Okay, so this is our enzyme. The normal enzyme, the reaction can happen. Okay, but if it happen, denature, if the temperature is too high, the enzyme will denature. So now it cannot fit. Okay, the substrate cannot fit into the enzyme. So now the reaction cannot happen. So the reaction will slow down or stop completely. Yeah. Okay. Okay. Yeah. So we can move this here. Okay. Yeah. So we can take a look at the graph. Okay. So this is what happened. Okay. So at low temperature. Okay. Okay. So when the temperature is low, the molecules, they have low kinetic energy. They are moving very slowly. So they do not form so many of the um, complex. Yeah. And then, so the rate of reaction is low, but at optimum temperature, they can form lots of products, okay, because they have more energy. But when the temperature too high, they denature, okay? So when they denature, the substrate cannot fit into the enzyme, so the reaction cannot happen, okay? Next, yeah, enzyme and pH. So the pH also have effect on the activity. Yeah, okay. So pH of a solution is either acidic or alkaline. So most enzyme, they will have optimum pH, okay? So they only work well in a certain pH. Usually it's around neutral. Example, okay, protease, the optimum pH is 2 in the stomach with hydrochloric acid. Yeah, okay. So we can see in this picture here, okay? So example, yeah, okay. So we can see this pepsin, okay, optimum pH of pepsin, okay, or protease is quite low, pH 2, okay. Salivary amylase is at 6.8 in the mouth, okay, yeah, around 7, okay. Catalase, pH 7.6, it happened in the plants, potato, example, okay, and then we have another one, okay, pancreatic lipase, okay. Pancreatic lipase, it works well at pH 9. Yeah, okay. So which means, okay, at the optimum temperature, the rate of reaction is highest, okay. When you go away from the optimum pH, the activity will be very low, okay. So when we have a wrong pH, it will slow down the enzyme activity, okay. But usually can be reversed if the optimum pH is restored, okay. So let's say we are here, the activity is low, but if we return back to the correct pH, the rate of reaction can increase. Okay. But if we have extreme pH, it can denature the enzyme. Okay. So if we have, like, let's say for pepsin, the pH is too high, again, denature can happen. Okay. It can damage the protein, the enzyme, it stop working. Okay, next. Yes, enzyme are specific. Okay, just now we also learn about that, but in the yellow box, in the um extended course, we need to explain in detail. Okay, so we can see the example here. Okay, hold on. You can take. Okay, yeah. So enzymes, they are very specific. Okay, they are very specific. Each chemical reaction is controlled by different enzyme. This is what we call high specificity. High specificity. So we can see here. Okay, so this enzyme, it can work for this substrate only. This enzyme, it can work for this substrate. This enzyme, it can work for this substrate. Okay, the shape must be right. Okay, if the shape is not right, it does not work. Okay, so which means like uh okay, so they must have the shape of the active site must be complementary to the substrate. Okay, so they can fit together well. So example, the active site of the protease will be different from amylase. Okay, so the protein, it cannot break down starch, and amylase cannot break down protein. So they are specific. Okay, so the enzyme molecule and substrate molecules, they will join together to form this enzyme substrate complex. Okay, so they will form this enzyme substrate complex, and the products will be released. Okay, so as we mentioned just now, they will form this. Okay, yeah, this okay enzyme substrate complex. Okay, yeah, they will form this. Only if they can fit together, they can form the enzyme substrate complex, and then they can get the product. If they cannot fit together, this cannot happen, and then the reaction also cannot happen. Okay. Next. Okay. Yes. Okay, so next we'll look at enzymes and temperature. Yeah, okay. So just now we already mentioned about this. Okay, so now we will go into more detail. Okay, so more detail to discuss what happened. Okay, so for the extended course, we need to know the detail of the explanation. Okay, yes. Okay. Yeah, so we can take a look at the picture. Okay. Okay. Yep. Okay. So basically, enzymes, they will work very slowly um at low temperature. Yeah. Okay. So at low temperature, they work very slowly because they have low kinetic energy and there are few collisions with the substrate molecules. Okay. So they don't meet the substrate molecules. Okay. So the reaction is slow. Okay. So as we increase the temperature, increasing the temperature, the rate of reaction will be increased because kinetic energy. Molecules, they are getting kinetic energy. So the rate of effective collision will be increased until the optimum temperature. So we will have the highest rate of reaction. Okay. And then above the optimum temperature, if we go over, okay, the reaction rate is start to decrease. Okay, so we can see it's going down. Okay, because the enzyme molecules permanently lose their shape at high temperature. Yeah, it deformed the active site. What happened here? Okay, so the active site changed the shape. So now the enzyme and substrate, they cannot fit together. Yeah, so you can see now they cannot fit together. So this is what we call denaturing. Okay, most enzyme will be denatured above 50 degree. Yeah, like above 50 degree, the activity it is zero. Okay, yeah, But enzymes are not denatured by low temperature. Low temperature is not denatured. No problem. It just slow down. Yeah, it will work again when the temperature is suitable. But if denature happen, it's permanent. Yeah, so it's irreversible. Okay. So next we will look at enzyme and pH. Okay. So we need to know like explanation. Okay. So how to explain it properly. Okay. Yes. So the enzyme will work best at the optimum pH at higher or lower pH. So if we have higher or lower pH, the enzyme will be less effective. And extreme pH, okay, if too much, okay, too high or too low, it can denature the enzyme. So the active site deform permanently, like what happened just now. So yeah, so it will, okay, deform permanently. So there will be no longer complementary fit between enzyme molecule and the substrate. Yeah, okay. So for the extended course, we need to be able to explain in detail. Okay, next. Okay, so now we will look at some sample questions. Okay, yeah, so you can take a look also. Here. Sample question. Okay, okay. Yep. So we have a biological washing powder. So means it has enzyme. Okay, so enzyme present. Okay. Okay, next. Explain why the presence of protease and lipase would make the washing powder more effective than ordinary detergent. Okay, why? Okay, so we can take a look the answer. Why it is more effective. Okay, that's because, okay, protease and lipase, they are enzyme, so they can break down the stain better than ordinary detergent. The protease will break down protein and lipase will break down fat to form products that are soluble. So the stain can be removed. Okay. Next, the powder should not be used in boiling water. We should not use boiling water. Why? Because boiling water, it will denature the enzyme. Okay. So they will not be able to break down stains. Okay. Next. Silk is a material made from protein. Explain why biological washing powder should not be used to wash silk. Okay, so we see here, do not use with silk clothes. Okay, why? Because in this powder, washing powder is got protein. Yeah, I mean protease. This protease, it would digest the protein in the silk, weakening the material and thus spoiling the clothes. Okay? Yeah, okay. So they have their advantage, also have their limitations. Okay? Cannot use for silk clothes. Okay. Next. Okay. Exam style questions. Okay, so we can take a look. Okay, next. Plan an investigation into the effect of pH on sample of enzyme amylase. Okay, so now we are given this enzyme amylase. Okay, we want to see, okay, which pH is suitable for amylase. Okay, let's say pH 2, 4, 6, 8, 10, something like that. Okay, so maybe 3, 5. Okay, 7, 9, 10, something like that. Okay, so we want to know which pH is suitable for amylase. So we're going to design a test to find out, okay, which pH is suitable for enzyme amylase. Okay, so first we know enzyme amylase is to break down starch. Okay, yeah, okay, so this will be our test. So we can look at the detail. Okay, 10-point question. Yeah, okay. So we need to think all the points that we can include. Okay. Yep. Okay. So we can talk about the apparatus and reagents like test tubes, something like that. Okay, and the reagents. Okay, independent variable. Okay, independent variable is what we are changing, the range of pH. So we can choose pH 1, 2, 3, 4, 5, 6, 7 until 10, 11, something like that. Okay, dependent variable, time taken for starch to be digested. Okay, so we are testing with starch because, okay, starch, it turns to maltose, okay, in the presence of amylase. Okay, so we just test with different pH and then we will see which one the digestion is faster. Okay, yeah, description, how variable should be controlled. Example, we'll use same volume of enzyme and substrate for each pH. We will maintain the same temperature. Yeah, okay, control water bath. And then we change the independent variable. Example, we can test pH 3 to 9. Okay, experimental procedure described. Okay, so example, we can say, okay, so example, we can take the starch solution, okay, and then we put the iodine, okay. So at first it is blue color, okay, and then we will put the okay, and then we'll put the amylase also, okay, and then we will put the um uh pH solution, okay, pH different pH solution, okay, and then we'll check example how long it takes, okay, how long it takes for the color blue to turn to brown. Okay, so we remove all the starch, right? So how many minutes it takes for the blue color to turn to brown color, something like that. Okay, then we will check. Okay, so we'll see at different pH. Okay, it will take the different amount of time for the reaction to be completed. Okay, other things we can talk about. Okay, risk and safety precaution. Example, careful when handling glassware, cleaning out spillage, and then if we have like acidic and alkaline pH, we need to be careful, wearing safety goggles. Okay, table for result drafted with suitable heading. So for example, we can have a table, okay? So we can have a pH and then time, okay? So different pH and then different time taken for the reaction to be completed, okay? And then prediction of expected result. Okay, so we know for amylase, okay, the optimum pH is 7, so we know the rate of reaction will be around pH 7, and then at the lower pH and higher pH there will be um uh lower rate of reaction, okay, because we know that pH 7 is the pH at the mouth, okay, where it is almost neutral. Okay. Okay, next. Okay, so here, uh, next question. Yeah. So 6 identical sample containing mixture of starch and amylase in water were kept at different temperature, and the time taken for the starch to be digested was measured. Okay. So here we are studying the effect of temperature. Yeah, different temperature and then different time for the starch to be digested. Okay, so we can see it makes sense. Okay, at 35 degrees, the time is low, which means it happened very fast. Okay, so here the time taken is long, and then here the time taken is long, so we are saying it's very slow. Yeah, okay, so the reaction is very slow. Okay, first part, plot a graph of this result. Yeah, so if we plot it carefully, we should get something like this. Okay, and yeah. So if we plot it properly, so we'll get something like this. Okay, so which makes sense. Okay, So at the optimum temperature, it takes the shortest time, okay, which means the rate of reaction is the highest, okay? Okay, question B1, describe how the time taken for the starch to be digested could be determined, okay? So how we can get the time, okay? So how we can get this time, okay? So what we are actually doing here, okay, is this. Okay, so first, we can take small sample of the mixture at regular time and test with iodine solution. Yeah, okay. When all the starch had been digested, the mixture will not turn blue-black. Okay, so that is one way to do it. Okay, next. Okay, B2. At what temperature was the starch digested most rapidly? At what temperature most rapidly? We know it's here. Okay, 35 degrees. Okay, yeah, 35 degrees. Okay, next one. Okay, describe the relationship between temperature and the rate of starch digestion. Okay, so we know the relationship. Okay, low temperature and high temperature not good. And then we have optimum here. Okay, so what can we say? Okay, how can we describe this relationship? We are saying that as temperature increase from 15 to 35 degrees, the rate of starch digestion will be increasing. From 35 to 50, starch digestion decreases. So optimum at around 35 degrees. Okay, so we basically describe the shape of this graph. Okay, yep. Okay, next. Similar sample was set up and kept at 10 degrees and 60 degrees Celsius. Okay, so now we add another 10 and 60. Yeah, okay. So what happened? Okay, was not fully digested after 1 hour. So this one is more than 1 hour, more than 1 hour. Okay, okay. Both of these samples were then kept at 35 degrees, suggest and explain the effect of the following changes, excuse me, in the temperature. Okay, so if we take the 10 degree, so we know this is low rate of reaction. This one is also low rate of reaction. Okay, but now if we go to 35 degrees, okay, now it can increase again, no problem. Okay, but if we take 60 degree to 35 degrees, no, because it's already denatured. Yeah, okay, so that's the difference. Okay, C1 and C2. Okay, so we'll look at the answer. Okay, so for C1, so if we take 10 degrees Celsius to 35 degrees Celsius, no problem. The starch would digest very quickly because exposure to low temperature will not affect the enzyme. It just slow down the action. Okay. But if we take 60 degree to 35 degrees, okay, the starch will not be digested at all because at high temperature already denature the enzyme. This effect is permanent. Okay. So now even if we take it to the optimum temperature, the reaction cannot happen. Okay. Next, question 3. Okay, so here we have adult female mosquito feed on the blood of mammals. Okay, no problem. They produce a protein digesting enzyme called trypsin. Okay, explain why adult female mosquito would need trypsin. Okay, so why? Okay, so we'll take a look. Okay, why that's the case. Okay, so the blood, it contains the proteins. So trypsin will be needed to digest them. Okay, so the mosquito use the trypsin to digest the protein in our blood so it can drink our blood easily. Okay, part 2. Okay, state the product that would be present in the gut of the mosquito if trypsin had been active. Okay, so we know our answer will be amino acid. Why? Because trypsin will break down the protein into amino acid. So in the mosquito stomach, there will be amino acid. Okay. Next. State one use of this product in the blood of an organism such as mosquito. Okay. So we are talking about amino acid. Okay. So why amino acid will be very useful for mosquito? Because uh, the amino acid, it can be made into proteins, okay, by the mosquito. So the mosquito can use it for growth, can use it to produce enzyme, okay, to make muscle or cell membrane, things like that, okay. So amino acids are very useful for the mosquito. Okay, next. Scientists found a way of introducing a hormone into the mosquito to switch off the trypsin secretion, okay. Yeah. So, just now the mosquito had the trypsin, so it can drink our blood easily. Okay. So now switch off. Okay. So now the trypsin is not there. Okay. How this treatment would affect adult female mosquito. Okay. So what happened to the female mosquito? Okay. Now the mosquito would starve to death because they cannot digest the protein in the blood they feed on. Yeah, okay. So now they cannot digest the protein, they cannot get the amino acid, so they will die. Okay. Enzymes such as trypsin are easily damaged. Suggest two ways of damaging the enzyme. Okay. So how it can become denatured. Okay, we know that. Okay, by high temperature or extreme pH. Okay, can denature the enzyme. Okay. Okay, cool. Yeah, so as I mentioned before, this one, very short topic. Okay, so we finish another one. Okay, so we finished unit 5 of the biology. Okay, yep. Okay, so this is also done. Okay, hold on. Yep. Wait. Okay. Yep. Okay, so this one, the textbook also done. Okay, so now we will try to work on the workbook. Okay, so this will also be very straightforward. Yeah, okay. So we can take a look. Okay. Very straightforward, the questions. Okay, let's take a look. First question. Okay. So give two key points for each term. Okay. Define the following terms. Okay. Definition of catalyst and enzyme, very straightforward. We know that. Okay. So catalyst, a substance that increases the rate of chemical reaction and is not changed by the reaction. Okay, we know that. Okay, enzyme, similar. Okay, so they are proteins. They are involved in all metabolic reaction and their function is biological catalyst. Okay, so they speed up the reactions, the chemical reactions in the body. Okay, next. Okay, so here we have enzyme control reaction and then we need to fill in the blanks. Okay, straightforward. So the starch, it will be the substrate. Okay, what we want to react. The amylase will be the enzyme and the maltose, it will be the end product. Okay, so like what we mentioned just now. Okay, yeah. Okay, what happened here? Okay, yes. Okay, so we have the starch molecule. We add the amylase. It breaks down into the maltose product. Okay, next. Okay. State two differences between a molecule such as starch and glucose. Okay, so what's the difference between the starch and glucose? Okay, so this is starch, this is glucose. Yeah, okay. So this is a very complex molecule. This is a simple molecule. Okay, so what will be the difference? what we can say. Okay, so we can say starch is a larger molecule, and then starch is insoluble. Okay, so starch is very large, so it's not soluble in water, but glucose is soluble in water. Okay, starch is a polysaccharide polymer. Yeah, we know that. Starch is a polysaccharide. Okay, just now we mentioned in the biological molecules. Okay, and glucose is monosaccharide monomer. Okay. Next, give two reasons why enzymes are important in all living things. Why? Okay. Why they are really, really important? Because enzymes will increase the reaction in cells and in systems, and then so that the processes they can happen fast enough to sustain life. Okay, so this is very important. Okay, next. Okay, so here we have a graph that shows effect of temperature on the rate of enzyme control reaction. Okay, yeah, so we are very familiar with this shape of the graph and then why it happened like that. Okay, so now on the graph, use the label line to add the following labels. Okay, so we need to label this one on the graph. Okay, so it's very straightforward. We know that. Okay, so we can take a look. Okay, easy, easy. Okay, yep. So this is what we expect to label. Okay, so we'll expect, yes, the reaction is slow here. Okay, and then the reaction is speeding up fastest here. And then here will be the optimum reaction rate, which is the fastest. And then here, denaturing is happening, okay, when the temperature is too high. Okay, next. Okay, predict and explain what happened if the reaction mixture at 10 degrees Celsius warm up to 30 degrees. Okay, we know that. Yeah. Okay. Okay, so if we have here, okay, 10 degrees warm up to 30 degrees. Okay, no problem. The rate of reaction will increase. The reaction will speed up because the substrate and enzyme molecules would move faster, gain more kinetic energy. There will be more collisions. Okay, in this case, it's not denatured. Okay, how about the reaction mixture at 60 degree, it returned back to 30 degrees. How about that? Okay, we mentioned just now, 60 degree, it returned back to 30 degrees, cannot, okay? Because the enzyme already denatured, okay? So there will be no reaction because all enzyme molecules have been denatured by the high temperature. Yeah, okay. Next, state three factors that affect the rate of enzyme control reaction. Okay, so we already know three factors. Okay, so first one is the temperature. Yeah, if we have different temperature, the rate of reaction will be different. And then different pH will also affect the rate of reaction. And then we also have one more, okay, that I will mention now. One more. Okay, the other one is concentration. Yeah, okay. So when we increase the concentration, okay, we can see the rate of reaction will happen. Okay. Yeah, because you see here, we have more enzyme substrate complex form. Okay. So when we have more substrate, we will have more enzyme substrate complex form. But when we have too much, can you see? Now the substrate is too much. Now the enzyme is saturated. Yeah, so now even if we increase the substrate, the rate of reaction will not change. Okay, but at the start, if we increase the concentration, the rate of reaction will increase. But when it is too much, it will stay the same because all the enzyme already used up, already saturated. Okay, so these are the three factors. Okay, what we mentioned just now. Yeah, okay. Temperature, pH, concentration of substrate. Okay. All three have an effect. Okay, next. Distinguish between a catalyst and an enzyme. Okay, so what's the difference? Okay, we know that. Okay, so enzyme is a protein, but catalyst can be inorganic molecule or element. Okay, so catalyst is everything. Okay, enzyme can be denatured by high temperature, but catalyst is not damaged by high temperature. Okay, enzyme has an active site, but catalyst does not. Enzymes are produced inside living organism. Okay, so these are all of the points that we can include. Okay, next, using appropriate terms, outline the action of enzyme on the substrate it breaks down. Okay, so what is actually happening there? Okay, so we can think about that. Okay, yeah, so basically we will focus on this. Yeah, okay, so basically this is what's happening there. Okay. Yep. Yeah. Okay. So, okay, outline the action of an enzyme. Okay. So what the enzyme is doing. Okay. Yeah. So basically the enzyme will be having the active site. Okay. So the active site will be having complementary shape to the substance is breakdown. So the shape it will be matching. Okay. So the substance breakdown is called the substrate. So what we want to break down is the substrate, what we want to react. Okay. So it will form the enzyme substrate complex. So after the reaction, the products will be formed. Yeah, okay. So we can see here, as we mentioned before, the enzyme remain unchanged after the reaction. So now it can continue to break down more substrate molecules. Yeah. Okay. So what we expect to answer there. Okay, next. Describe action of digestive enzyme on a large insoluble food molecule. Yeah, okay. So this is what we're talking about, like the this one example, like the starch. Okay. Yep. Okay. So example like the starch. What happened? Okay. So the food molecule is the substrate. Okay. So example this starch. The enzyme has an active site. Yeah, like what we mentioned before. Combine temporarily with the substrate to form enzyme substrate complex. Okay. The bonds are broken in the substrate molecules. So these bonds, we are breaking some of the bonds. Yeah. Okay. So like this one, we are breaking this bond here. Okay. The products are formed. Okay. Now we start with a food molecule that's insoluble. Okay. Like the starch. And then in the end, we get small soluble molecules like the maltose. Okay. Okay. Next. Uh, okay. Explain why digestive enzyme usually works on one type of food molecule. Yeah, okay. So this is what we mentioned just now. Okay. Yeah. Okay, what we mentioned just now. Okay. So basically, it's very specific. Yeah, like this one. Okay. So like let's say we have this one. Starch is only for amylase. Okay. And then we have protein is only for protease and so on. And then we have fats is only for lipase. Okay. It's very specific. Okay. Why is very specific? Because of the shape. Okay. Enzymes have a specificity. They have complementary shape. Okay. To the substrate molecule they work on. So the active site and substrate, they can fit closely together to allow the reaction to happen. Yeah, okay. So basically that's the one. Okay. Yeah. Okay, now. Okay. This is good. Okay, so basically we finish everything. All the notes and all uh the workbook as well. Okay, so unit 5 is completely done. Okay, so this is good. Okay, unit 5 is completely done. Okay. Okay. Yep. Completely done. Okay. So now what should we move on? Okay. So basically we finish everything. Physics, forces and momentum, biology, biological molecules done, enzyme done. Okay. So now we will move on to chemistry, unit 4. Okay. Stoichiometry, chemical calculations. Okay. So this is all about chemical calculations. So we still have around like 14 minutes. Okay, so we're just going to get started on this stoichiometry. So this is a big topic. So we're going to get started and then try to uh, uh, make some progress, and then we can continue next time. Okay, so as I mentioned before, your mom informed tomorrow no class. Yeah, so tomorrow no class. So we will no, yeah. So tomorrow no class. So 15, today is 15. So tomorrow 16 no class. So we continue on the 17. Yeah, okay. So we'll continue on the 17. So now we just quickly get started on this chemistry unit 4, and then the rest we will continue on the uh Thursday. So tomorrow there will be no class. Okay. Okay, let me open. Okay. So now okay, so now we move on to this chemistry. Okay, unit 4. Okay, okay, chemistry unit 4. So we'll be in progress. Yeah. Okay, so this is a big topic, so it will take some time. Okay. Yeah, so I already sent the notes to you some time ago, so you can take a look. Okay. Yep. Unit 4. Yeah. Okay. So this is a big topic. This is all about calculations. Okay, so need to memorize quite a bit of formula here. Okay, yeah. So these are the key terms that we need to know. Okay, so we'll come into this later. Okay, relative atomic mass. Yeah, okay. So relative atomic mass is just talking about the elements, okay, in the periodic table. Okay, yep. So relative molecular mass is for covalent compound. Okay. So when we're talking like covalent compound like water, CO2, and SO2, like C2H6, something like that, all of these covalent compound will use molecular mass. Relative, okay, formula mass is for ionic compound. Okay, so relative formula mass like NaCl, okay, and then NaOH, okay, MgCl2, all of these, okay, it's just talking about the relative formula mass. Okay, so now our first practice is to calculate MR. Yeah, okay. So it's for the calculation of MR, which is very, very easy. Okay. So we just need to have the periodic table. Okay. So okay, so let me open the periodic table. Let me open one periodic table. Okay. 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 Yep. So we will be provided with a periodic table in the exam. Okay, so we can use this periodic table for our calculation. Yes. Okay. So this calculation is very, very easy. Okay. So let's say you want to calculate iron sulfide. Okay. Iron sulfide is Fes. Okay. So we can see this is 1 Fe, okay, plus 1s. Okay, so we can go Fe. Okay, So we are focused on the mass, so we will focus on this number, not this one. This is proton number, so we'll focus on this one. 56 for Fe, and then sulfur is 32. Yeah, okay. So we just simply plus them together. 56 plus 32 equals to 88. Okay. So the Mr for the iron sulfide, okay, Mr for iron sulfide will be equal to 88. Okay, just like that. Okay, so if we want to put the unit, we just put gram per mole. Okay, just like that. Okay, so we have a few more examples. Yeah, okay. CO2, N2O, C4H10, PbNO3, 2. Okay, Al2SO4, 3. Okay, and then this relative atomic mass we can get from the periodic table. Okay, so let me just copy this. Okay, yeah, okay. So this part is very, very easy. Yeah, okay. Yes. Okay. Okay. Uh, yeah. Okay, so first, CO2. Okay, CO2 is 1 carbon plus 2 oxygen. Okay, so it's 12 plus 2 times 16. Yeah, so you can see here, oxygen is 16. Yeah, okay. So all of the relative atomic mass is provided. Okay, so it's easy. So we just calculate like that. N2O, 2 nitrogen plus 1 oxygen. Okay, so 2 times, okay, nitrogen is 14. Okay, plus 1 oxygen. Oxygen is 16. Okay, so just like that. So that will give us 44. Okay, C4H10. So we have 4 carbon plus 10 hydrogen. Yeah, okay. Carbon is 12. Okay, hydrogen is 1. Okay, 10 times 1. Okay, so that will give us 58. Okay, so very, very easy. Okay, so now, okay, more example. Okay, let me just move this. Okay, okay. Oh, yeah. Okay, so now, PB, okay, lead nitrate. Yeah, so lead nitrate, we need to be very careful. So lead is outside of the bracket. So lead is just 1. 1 lead. Okay. Nitrogen, we need to multiply outside the bracket. So it's 2 nitrogen. Oxygen, 3 oxygen, multiply the bracket. So that will be 6 oxygen. So lead is 207 plus 2 nitrogen plus 6 oxygen, like that. Okay, 331. Okay, just like that. Okay, next. Al2SO43. Okay, so this part we need to be very, very careful. Okay, so we can see here 2 aluminum. Okay, and then sulfur. 1 sulfur multiply the bracket, so there's 3 sulfur. Oxygen 4 times 3 outside the bracket, 12 oxygen. Yeah, okay. So aluminum, okay, we can see here aluminum is 27. Okay, sulfur is 32. Okay, okay, so it's 2 times 27. Sulfur is 32. Okay, 3 sulfur, 32. And then 12 oxygen. Oxygen is 16. Okay, plus all together. So that gives us 342. Okay, so this is the most basic calculation for the stoichiometry. Easy, easy. We just need the periodic table. Okay, next. This is the last one. Okay, so we do this one and then we stop. Okay, we end. Okay, next. We can convert relative atomic mass and relative molecular mass into actual mass by adding the mass unit. This allows us to work out the masses of substance needed to produce in reaction. Okay, example. Yeah, let's take a look. Calculate the mass of sodium chloride that can be produced when 4.6 gram of sodium burned in excess chlorine. Yeah, okay. So now, okay, let me just copy this equation. Okay, this equation. Okay, so we can put here. Okay, equation. Yes. Okay, so this is our equation here. Okay, so okay, what we want to calculate. So we want to calculate if we put 4.6 gram of sodium, okay, how much sodium chloride, okay? Okay, so this one, if we put 4.6 grams, so this one will be how many grams? That's our questions, okay? So from the periodic table, we can get sodium chloride is 1 sodium, 1 chloride. 23 plus 35.5 is 58.5, okay? MR of sodium chloride is 58.5, okay? Excess chlorine means it's more than enough to react with all the sodium. So we don't care about chlorine. Okay. So now we see carefully. Okay. From the equation, we can see 2 sodium will react, will produce 2 sodium chloride. 2 sodium produce 2 sodium chloride. Okay. So which means now, okay, um, 2 sodium, which is 46 grams, okay? 2 sodium chloride will be 117 grams, okay, of sodium chloride, okay? So, which means, okay, if we put the 2 sodium, okay, which is 46, we will get 2 sodium chloride, which is 117. Okay. But what if we just put 4.6 grams? Okay. 4.6 gram can give 117 grams. What if we just put 4.6 grams? So this one will be divide by 10, divide by 10. Okay. So which means 11.7 grams. So in the end, we will get 11.7 grams, okay, of the sodium chloride. Okay, so which means if we put 4.6 grams of the sodium, we will get 11.7 grams of the sodium chloride. Okay, so that's one calculation that we can do. Okay, so the time is up. So next time we will continue. Yeah, okay. So we are up to this point here. Next time we will continue. It's a big topic, so we need lots of practice. Okay. So yeah, so our progress is here. Okay. Yes. So next class, which is on Thursday. So tomorrow no class. On Thursday we will continue this chemistry. Okay. Yeah. So if no other question, we will stop here. All right. Okay. Thank you. Bye bye. Bye bye.